Hello, BISC130. This is the beginning of recorded lecture 5.5, the last recorded lecture of the quarter. So we're still in the chapter on biotechnology. We began this chapter in the previous recorded lecture, and I introduced the overall process of molecular cloning. And last time we talked about, this first step of getting our insert or our gene of interest. Now we have to talk about these plasmids and how we get them. It's labeled a vector here, but they're also known as plasmids. How do we get these things? Well, how to get plasmids. The whole reason we're using plasmids in the first place is because this is something that bacteria understand. We don't want to just throw our gene of interest at the bacterial cells. They're not going to recognize it as something important. They're just going to break it down and use it for energy. But if we put it into a plasmid, it's something they're going to be able to pick up and transcribe and translate and recognize. So we get these plasmids just naturally from bacteria. Again, this is something that they're used to. There are many plasmids that are naturally occurring in many different species of bacteria, and without getting bogged down into the details, it's easy to purify these plasmids from those bacteria. The only other thing I want to note about these plasmids is that, naturally, we just pick these up straight from bacteria, but over the years, over the decades of doing molecular cloning, scientists have tinkered with these things, modified these plasmids to be, again, I'm being kind of vague here because this encompasses a lot of different things, modified to be better for molecular cloning. So there are tons of plasmids out there that exist. This is a table from some random scientific journal article and showing different strains of E. coli that they used and the different plasmids that just this one study, just this one journal article used like half a dozen different plasmids, all with very delightful names, PKD4, PSE380, all this stuff. Don't memorize any of this stuff, but this is just kind of showing you the different kinds of things that can be done to these plasmids that can make them just more convenient for the overall process of molecular cloning. So we get them from bacteria, we've modified them over the years, but yeah, getting these plasmids is pretty straightforward. So we've got our plasmid or vector, we have our gene of interest, now we got to put the two together. And this is probably the most complicated part to try to explain in this overall process, because we're going to have to cut open this plasmid and we're going to have to glue in this gene to create a slightly larger plasmid. So how do we cut and how do we glue? How do we cut the plasmid open? How do we seal in our gene of interest, also known as an insert in this context when we're putting it into a plasmid? Well, to start by talking about the cutting. To do the cutting, we're going to use things called restriction endonucleases, also known as restriction enzymes. It's used interchangeably. I think they're usually called restriction enzymes, but the technical term for these things is restriction endonucleases. So let's talk about restriction enzymes a bit. Sort of a side note, side tangent here. Restriction enzymes. There are a ton of these that exist, and here's another table that I definitely do not want you to memorize, but this is just showing the A through B part of the alphabet, so there's even more than just this. There are tons of these enzymes that exist. They all have a specific sequence at which they cut. So this AAT2 cuts at GACGTC, and all these other different restriction enzymes have other sites at which they cut. So there are many of these that exist. They're all different in their own special ways, but each one has a unique sequence at which it cuts. So they're not cutting randomly, they're cutting at specific sequences. Now, there's something about these unique sequences. So they cut DNA at a very specific sequence. They cut at what is called an inverted repeat palindrome sequence. What a phrase. Inverted repeat palindrome sequence. What is an inverted repeat palindrome sequence? Well, I think the best way to understand what this is to look at an example. You do not need to memorize this specific example sequence, but it will show us what an inverted repeat palindrome is. this is two sequences, of course, because this is double-stranded DNA we're talking about. And this is GAATTC on one strand, and the other strand, C's pair with G's, A's pair with T's, so the other strand is CTTAAG. What's so special about this? Well, let's read this first strand again. GAATTC. Now, let's read the opposite strand, but we'll read it backwards. GAATTC. It's the same sequence. So GAATTC is up here, GAATTC is down here. My best way of trying to describe this is an inverted repeat palindrome sequence is a sequence in which opposite strands read backwards from one another. Not every random snippet of 6 nucleotides in a sequence is going to do that, but with a very specific sequence, you're going to have the two strands, if you read them in opposite orders, are going to read the same thing. GAATTC on one strand. If you read the other strand and you read it backwards, you'll find the same sequence, GAATTC. That's an example of an inverted repeat palindrome sequence. And yeah, like I said, these restriction enzymes specifically cut at this kind of sequence, inverted repeat palindrome sequences. But it's not just the specific sequence that they recognize. It's within that sequence they make very specific cuts. Restriction enzymes cut between two specific bases within that sequence. So let's go back to this again, the GAATTC, the enzyme that recognizes this sequence. I think it's BAMH1 or ECHO-R1. They all have names like that. The enzyme that recognizes GAATTC cuts between the G and the A. So it looks for GAATTC, and then it's going to make its little cut specifically between the G and the A within the context of this sequence. That's what I mean when I say they cut between two specific bases in their recognition sequence. This is important because if we are going to cut DNA, remember DNA is double-stranded, it's important that we cut both strands because we want to actually break apart this molecule of DNA. If you only were to cut one strand, the DNA would not come apart. You have to cut it twice in order for this to actually come apart here and open up. And this is why the inverted repeat palindrome sequence was so important. If you are cutting between G and A, every time you see GAATTC, if both strands have GAATTC, that's going to ensure that every time your special sequence comes up, you're going to cut both sides of it. You're going to cut both strands of this double-stranded DNA. That's only going to happen if your recognition sequence is an inverted repeat palindrome sequence. We want to be able to cut both strands. Importantly, these two cuts are staggered. They are uneven. So these restriction enzymes make uneven cuts here. Again, sort of staggered. We can see it here. This results in something called sticky ends. Sticky ends are short sequences of single-stranded DNA. Try to say that 5 times fast. But in order to look at these sticky ends, we have to look at what happens after these cuts have been made. So once this cut has been made and the G has been severed from the A, this phosphodiester bond, if you remember that term from many chapters ago talking about DNA, this phosphodiester bond has been severed. This phosphodiester bond has been cut apart. The only thing holding this together are the hydrogen bonds between these nucleotides. And, 4 sets of hydrogen bonds, as it turns out, is not particularly strong. So once these cuts have been made, the thing is going to fall apart. The AATT and the TTAA, those are our sticky ends. Again, short sequences of single-stranded DNA. They are called sticky ends because, well, they're the end of the DNA, and they're sticky. DNA does not like to be single-stranded. It wants to be double-stranded. This AATT is not very happy. These A's are looking for T's. The T's are looking for A's. They want to stick to something. They want to find partners. That's why they're called sticky ends. So this was just showing, this one sequence in isolation and what happens when it is cut apart. Remember, this sequence is not just going to be 6 nucleotides floating around. This is part of a larger sequence, a plasmid. So yeah, here's that same thing. Oh, it was echo R1, the GAATTC. And yeah, there's more sequence before it. There's more sequence after it. You could imagine this being as part of a very long sequence of DNA within a plasmid. Here's the cut being shown. And here's the separation happening. And again, these are the sticky ends, the TTAA, the AATT. Importantly, when we do this cutting, when we select which restriction enzyme out of, the massive book of different restriction enzymes that exist, we need to pick a restriction enzyme that is only going to find its cut sequence once within the plasmid. We want to choose a restriction enzyme that will cut the plasmid at one site. Again, it's going to cut both strands, but it's only going to find itself one cut site. When we do this, when we pick a restriction enzyme correctly and we treat our plasmid with this restriction enzyme, it's going to look like this. So yeah, this is vector. Remember, vector, plasmid used kind of interchangeably within this context. And yeah, we see 2 different strands here, double-stranded DNA, sort of color-coded. And yes, we treat it with a restriction enzyme that finds its site once, and yep, it's going to cut open this plasmid just like this. You can see the little sticky end here, you can see the little sticky end here, and yeah, this plasmid has been cut once. Meanwhile, we need to do something to our insert, to our gene of interest. We're actually going to cut both ends of the insert, the gene of interest, with the same restriction enzyme. So this is going to involve 2 cut sites, one at either end of the insert. I'll show an image of this in just a second, but I wanted to describe it first. Because we are cutting both ends of the insert with the same enzyme, that's going to give both ends of the insert the same sticky ends as the plasmid. So let's visualize this. Here's our insert, our gene of interest. We're going to cut this with restriction enzymes. That's going to produce, here's the portion of the gene we actually care about. These are a couple of edges that we don't really care about. But yes, again, this is too zoomed out to see the GAATTC or whatever, but you should notice the sticky end right here, the sticky end right here. Because this restriction enzyme that was cutting the insert is the same restriction enzyme that was cutting the plasmid, this overhang sequence right here is going to be complementary to this overhang sequence right here, which means they're going to want to stick to one another. This is, AATT, this is TTAA. They're going to be complementary. If you throw in this cut insert in the same little tube as this cut plasmid, they're naturally going to want to come together and have their sticky ends stick to one another. So we're done talking about the cutting. Now we're going to talk about the gluing. So we are going to mix the cut plasmid and the cut insert. They're going to bind together at their sticky ends, giving us something like this. However, this is not a particularly strong connection here. The only thing holding together right here, this sort of combo of plasmid and insert, is about 4 sets of hydrogen bonds between 4 nucleotides. 4 sets of hydrogen bonds between these 4 complementary nucleotides. Just putting these together, yes, they will stick, but there's going to be something missing. This is going to be not a strong connection, as I put it. because there's a phosphodiester bond missing on each of these connections. All the nucleotides are going to be there in this sequence, the A's and the T's, the C's and the G's, but there's going to be sort of a gap. You cannot see it here, but there's a little missed connection between the red and the dark blue here, a missed connection between the light red and the light blue here, and a missed connection here and a missed connection here. All the sequence is present, but there are going to be these gaps, these missed phosphodiester bonds. This slide might look familiar to you. We had this exact same problem of missed connections way back when we were talking about DNA replication. After DNA polymerase I filled in this gap with DNA, after removing the RNA primer, we had this exact kind of missed connection. And the enzyme that dealt with this problem is the same enzyme that we're going to use here. It's an enzyme called DNA ligase. DNA ligase can take this sequence of nucleotides and this sequence of nucleotides with a missed connection and fill that in, glue everything together on a much more permanent basis. So after we've done this cutting and after we've done with this mixing, we're going to add DNA ligase. It is going to covalently seal the plasmid and the insert together, making a very strong connection here. And now this is a larger plasmid that incorporates our gene of interest. So again, this cutting and gluing is admittedly, I think, the most confusing part to try to explain. But again, you can go back and watch this again, whatever. I've gone through the process of cutting, the process of mixing, the process of gluing. Now we're back to this sort of overview here. We have our plasmid, which now contains our gene of interest. Our next step sounds difficult, but is actually pretty straightforward. Our next step is we have to take this plasmid and we have to get it into a bacterial cell. Not to scale here. This is more to scale. Well, plasmid here inside of our E. coli bacterial cell. This should sound difficult because DNA is a big, bulky molecule, and there's the plasma membrane it has to get through, and the cell wall. Remember, bacteria have cell wall. This process should be difficult, but it's not. So, how to get plasmid into bacterium? Well, this process is called transformation, and it involves a lot of tedious steps that I don't want you to memorize. You're incubating things at specific temperatures, and you're spinning them, and you're putting them on ice, at a different temperature, and you're spinning. It's a lengthy process that's not particularly interesting, but by messing with the temperatures, heating up and cooling down and adding specific chemicals, we can get this plasmid to get into the bacterial cell. So we're not memorizing all the different times and temperatures here. All I want you to know is that this process is called transformation. Transformation is the name of this process that gets a plasmid into a bacterial cell. That's all I want to say about this step. So now we have our bacteria that have the plasmid, which contains the gene of interest. Now we have to grow these bacteria. So how to grow bacteria? Well, this particular slide looks like it's growing them on a petri dish, little colonies of bacteria on a petri dish. But if we want to grow lots of bacteria, and again, the whole point of this technique, the whole point of molecular cloning was to get lots and lots and lots of protein that is encoded by this gene of interest. If we want lots of bacteria, we're not going to grow them on a little dish. If we want lots of bacteria, we are going to grow them in liquid culture, 100, 200, sometimes even a liter of this stuff. So this is liquid media, looks kind of like urine, but really this is perfect bacteria food. It's got all the vitamins and nutrients and amino acids and other things that these bacteria need to grow really, really fast. In fact, this is this is media just that's been straight made. If you add a tiny little pinprick of E. coli, an invisible microscopic amount of bacterial cells to this bacteria food, and you let it grow overnight, less than a day, just overnight, you will come back into the lab the next morning, and that media is going to be absolutely cloudy with bacterial cells. That's what the color change is. This is chock full of bacteria. So if we want them to grow optimally, we have to put them in conditions that they're used to for optimal growth. Remember, E. coli are used to living inside of a human. Even the non-pathogenic varieties like to live in our large intestine. So you can grow this stuff at room temperature, but if you really want them to grow fast, you will put them inside of an incubator that is going to warm them up to human body temperature. Then they'll grow at their optimal speed. The other thing that's going on inside of this incubator, I had an animated GIF, but it was too annoying to stare at. But this incubator is going to shake back and forth. It's going to move around in sort of a circular motion. It's just going to keep this liquid aerated. These things, they're living things. They need oxygen in order to do cellular respiration. So by sort of gently shaking these flasks, it's going to ensure that this liquid has oxygen dissolved in it as well. So how do we grow bacteria? In liquid media, in a heated, shaking incubator. It's heated to human body temperature, because again, that's what they're used to, and it's shaking in order to keep E. coli in liquid. The whole reason we're using E. coli as our bacteria is because they grow quickly and they're really easy to take care of. Importantly, as they are growing, as they are dividing, they will be expressing that gene of interest on their plasmid. They will make lots and lots and lots of that protein of interest as they grow. Finally, well, I guess not finally, we need to break open these cells, and then we need to purify our protein from everything else. So our next step is breaking open these bacterial cells to get the protein out. How do we do that? Well, there are actually several different ways to do that. So one way to break open bacterial cells is just chemicals. And there are many different chemical products that exist. They're all going to mess with the cell wall in one way or another. That's going to cause a rupture, and all the protein and other stuff is going to leak out. So how to break open cells? Chemicals, which will chemically, again, I'm being vague because there are a lot of ways to do this, chemically break down the cell wall. That's going to liberate the contents of these cells. Or we could use something called a sonicator, which, I have a Sonicare toothbrush over here. It's not a coincidence that they both have Sonic in the name. They operate on pretty much the same principle. This, toothbrush is going to vibrate with enough force to, move off bits of food and stuff from your teeth. This sonicator is going to vibrate so intensely that the physical movement of this metal tip is enough to break open those bacterial cells. You won't be able to see this moving. That's how small its movements are. It makes a horrible noise if you ever have the pleasure of being in the same room as one of these operating. But yes, it's sort of mechanical force and pressure that's going to rupture open the cells that are near the tip of this sonicator. So chemicals, capital, or something called a sonicator. It vibrates with enough force to mechanically rupture cells. Again, we're not treating them with chemicals in this technique. It's mechanical rupturing. Or there's a third way of doing this called a French press. Oops, wrong French press, Google image search. This is making coffee. But actually the French press in a lab operates under pretty much the same principle. Here you take your coffee and your water and you use some force to push it through small holes that are going to press it through. The French press in a lab setting still uses force, but it's industrial, very large amounts of force to, again, if little diagram over here. You've got our cell suspension. You've got an enormous amount of pressure pushing down on these cells and the liquid they're in. And there's a teeny tiny little hole here at the bottom, and the enormous force being pushing pushing these cells through the tiny little hole is enough to mechanically rupture open these cells. Again, it's an enormous amount of pressure on these cells. It's going to break them open as they pass through this tiny little hole, and then you can collect what comes out at the bottom. So the French press technique is another thing that doesn't rely on chemicals, it's relying on force. It forcefully pushes cells through a small hole, shearing them open. Any of these three techniques are going to be able to break open bacterial cells. Sometimes multiple of these are used, one after the other, to make sure that these cells are really ruptured open, because you're not going to be able to get your protein out if you don't break the cells open first. Okay, now we move on to the final step of molecular cloning. We have broken open these bacterial cells, they've released all their guts. We need to get our protein of interest away from all the other stuff. How to purify this protein of interest, because it's not, they've been making our protein, but they've been making a bunch of other proteins. Again, these E. coli are living things. They're building a lot of proteins that they need to stay alive, and they're also making sugars and lipids and DNA and all sorts of other macromolecules. All we want is protein, and all we want is our protein. How do we get this in pure quantities? Well, once again, this is one of those things where there are many methods to make this happen. Unlike the rupturing of cells, I'm not going to give you many examples. I'm just going to give one example of how protein purification can work. My example of protein purification is something called size exclusion chromatography. Complicated sounding name. It's not as complicated as it may sound. So size exclusion chromatography is a technique that's going to separate all the different proteins based on what else? It's right there in the name, based on size, based on how big they are. Again, there's going to be a bunch of proteins in this lysate, in what you get out of after you've broken open these E. coli cells. You know how big your protein is. If you could separate them all out based on how large or small they are, you're going to be able to get your protein. So how does this work? Well, this uses a column, which they vary in size. Some of them are a little bit bigger than a roll of pennies. Some of them are a foot or two long, and these look like they're just white columns. Really, these columns are filled with teeny tiny microscopic beads, and I'll show you what those beads are like in just a second. But you're going to pour your solution, the guts of these broken open bacteria, you pour it into the top of the column. It's going to drip down, sometimes by gravity, sometimes with a little bit of pressure applied, and everything will drip out of the bottom eventually to be collected by this machine. But they're not all going to drip out at exactly the same rate. They're going to drip out differently at different speeds based on their size, like I said. So what's going on with these beads? Well, let's zoom in on one of these columns and see. These are the beads, these little black circles here, and these are our proteins starting at the top. So again, there are going to be lots of different proteins of lots of different sizes, but to simplify things, let's look at what happens when you have just two proteins, a big one and a small one. So the proteins start at the top, they drip through, and the big ones are going to flow through quickly, whereas the small ones are going to move slowly. Wait a second. That sounds backwards, right? Usually big things are slow and small things are fast. Why are the big ones moving through fast and the small ones moving through slowly? Well, to understand why this is happening, we have to zoom in even further to look at the structure and the texture of these little beads, because they're not perfect little circles. These beads are highly textured. There are lots of cracks and crevices and nooks and crannies of varying sizes. So what happens as these proteins pass over these beads is that small proteins and other small things are going to get stuck in these cracks and crevices. Not permanently stuck, but stuck enough to where they're going to spend some time in this crack, and then they're going to fall out, and then they'll get stuck in another one, and then they'll fall out, and they'll get stuck in the crevice of the next bead, because again, they're going to pass by lots and lots of beads as they make their way through this column, whereas larger molecules or larger proteins are maybe they'll get stuck in some of the bigger cracks, but they're going to move through more quickly because they're not spending as much time getting stuck. So again, this is a simplified example of, small protein, big protein, but everything else in the middle is going to come out in the middle. The biggest of proteins will drip out first because they're not getting stuck in hardly anything at all. The smallest of proteins are going to drip out of the bottom of this column last because they're getting stuck in every single bead that they bump into. And any medium-sized proteins are going to drip out somewhere in the middle. They get stuck in some of the bigger crevices, but not others. And so all these proteins you start out with at the top, they all are going to drip out eventually. Nothing is getting permanently stuck. Nothing is getting destroyed here, but they are going to drip out of the bottom and be collected by, a fancy little fraction collector like this, according to how big they are. So, in summary, in size exclusion chromatography, we're going to run this protein solution over a column of porous beads. Small proteins get caught in the porous, they travel slowly. Large proteins pass through unhindered, not getting caught in all these pores, and they're going to travel quickly. Again, sort of unintuitive, kind of backwards, but hopefully you understand why this is true. All proteins drip out eventually, and we can find our protein of interest in the appropriate fraction according to its size. You should know how big your protein of interest is. Of course, you will confirm its presence in the appropriate fraction. But yeah, you're going to find it either coming out fast or coming out later, depending on how big it is. And there we have it. Now we have our protein. We've got a lot of it, and we have a very pure version of this protein. We can use this for experiments. We can also use this in a medical context. So I didn't really mention this when we were going through sort of our overview here. The big picture purpose of all this is to get protein. Yes, it can be done for scientific experiments. This could also be used in medicine. Insulin is a protein that is normally made by the human body. If you have type 1 diabetes, your body is not making enough insulin. And back in the day, sorry, I don't have any slides for this rambling story here at the end. Back in the day, people with type 1 diabetes had to inject themselves with horse insulin or pig insulin, purified from the pancreases of those animals. It had some pretty bad side effects. Nowadays, if you're type 1 diabetic, you inject yourself with human insulin. And we didn't have to rob any graves or grind up any human pancreases to get that human insulin. We have lots of pure human insulin to use because of molecular cloning. Gene for insulin, put it in a plasmid, put it in bacteria, grow those bacteria, break them open, purify that human insulin protein from everything else. Now we've got that protein we can use as a therapeutic. So again, the reason why I chose molecular cloning as our topic for this entire biotechnology chapter is how widely this technique is used anytime you need protein. So anyway, that brings us to the end of this chapter, the end of this lecture, and the end of this course. So this is our cutoff for exam number 5. This is the end of everything. I hope this was a good capstone because it referred back to topics from many chapters throughout this quarter. But yeah, this is the end of recorded lecture 5.5.